Thank you very much, Peter. The introduction was so short that I thought that I will put my CV there to begin with. I want to thank Betty for inviting me here. This is the first time in Evergreen, and I have enjoyed a lot. I have met some old friends and I have made some new ones. I'm very happy to be here. So, to the business. I have 3 topics and I think I have time to cover 2. So, the first 2, that's why the last third is in parentheses. So, some words about the phage therapy initiative which we have started in Finland. The first aim here is that we would like to set up the phage therapy laboratory in Finland, and so this is a very modest funding that I received for this and next year. I can hire 1 master student to do some work on this, and our first aim is set up a phage collection against the SKP bacteria. And so we have received 10 isolates of each of these SKP isolates from the local clinical microbiology laboratory, and the long-term goal is to make a clinical study of wound infections. Then, if we get permissions and all things set up. So, very shortly, this is how we are now working. We started in March. We isolate phage and do some characterization to begin with. So this is the enrichment procedure, and we follow that until so that we purify the phages and make stocks, isolate DNA, do some restriction digestions, select those which look different, and then do host range analysis. So this is how we then pick up some of the plaques there for purification and so on. We thought that we would like to cover more or less the whole Finland, so we sent emails to different cities in Finland and asked them to send sewage to us, and that's what they did. So here you can see this SKP, which bacteria it covers. And then this is the list, the present list of the phages that we have isolated from different city sewages. If you were here Sunday morning and heard Andrew to tell that what should not be present in the phage name, so we don't have phi there, we put F in the beginning. And then we have the city and then the bacterium and then the number of the phage. Start trying to keep some kind of order there. Out of these sewages, we didn't find any phage against Acinetobacter and Pseudomonas and Staphylococci. So then we thought that, and after exchanging some emails with Betty, that perhaps we could use hospital sewages instead. So the next map is the map around Helsinki, and here we have contacted several hospitals, and they are very cooperative, so we got sewages from all these different hospitals, and then started to do the phage isolations from there. And as you can see here we have Aeruginosa, Acinetobacter, and this morning, yesterday evening, I received email from my student that she has also isolated now 1 Staphylococcus aureus. So in this way we have now isolated several phages and then studied a little bit of the host range. So here with the green pluses is the strain that was used for enrichment in the beginning, and as you can see, quite expected, this is this strain for some reason only picks up phages which are specific for itself, a little bit reaction with others, and you can see that here we have E. coli phages, and here we have some more broad host specificity. This is Enterobacter cloacae, not the kind of the message here is that we need to use many different strains to pick up phages so that it would cover the whole range of these isolates. So here we have some summary of these phages which we have isolated, altogether 53 phages, and now we have also at least one isolate against aureus. Before we started with this SKP, we took also some Yersinia enterocolitica and could isolate phages which are specific for serotype 9, 8 and 3. So this is the summary of this phage hunting. We have altogether 62 phages now, and they kind of are distributed like this. The next step here is that we will have all the phages sequenced and analyzed for the possible use in phage therapy later on. And because we want to isolate more phages to get a better covering collection. So this is Anu Wiklund, who has now worked half a year and has done a lot of good work. Okay, so now to the second topic, Yersinia. Yersinia is the genus in Enterobacteriaceae, if you don't know that, and it has 3 pathogenic species: Enterocolitica, Pestis, Pseudotuberculosis, and Rückeri. Yersinia rückeri is causing disease in fish, and Pestis is the causative agent of bubonic plague. Pseudotuberculosis and Enterocolitica cause gastroenteritis, and the other species which are here listed, they are more or less environmental non-pathogenic strains. So Pestis, Yersinia pestis causes the bubonic plague and pneumonic plague. Yersinia enterocolitica, Yersinia pseudotuberculosis cause gastrointestinal infections. And I have studied these bacteria because they are very good system model for studying bacterial pathogenesis and virulence. Some words of the evolutionary relationships between these ones. It is thought that long time ago there was an ancestor for Yersinia, and there was a split to Enterocolitica-like bacteria and pseudotuberculosis-like bacteria, from which Pestis has evolved perhaps even less than 5,000 years ago. And it is believed that it has evolved in China, somewhere in China. So here is a list, complete list of the sequenced bacteriophages which have been annotated in my lab, and today I will tell a little bit more of this Yersinia Enterocolitica specific phage R137. I have another red highlight here is YerA41. This is the fish pathogen Yersinia rückeri, and this NS means that it has not been able, we have not been able to sequence it, even with the new sequencing technology. So we heard about a similar case that some phages are really, some DNAs are really not sequenceable, and this is one of those. So there's still something to do for the sequencing technology. So here is the phage R137. Now I would like to know who has really read this paper that we published last year. Yeah, 3. Very good. So then it is not useless to describe what is going on here. So here is the wrong nomenclature with a phi. But this is what we had to, because this phage had been isolated earlier. As you can see, here is some history of the phage. It was isolated 89 in Turku, from the sewage in Turku, and I used a host strain R1, and so the name R1 comes from there. from there, and because the bacteria were grown at 37, so that is the last part of the name. So R137. We have also phage R1RT, which when we are growing the bacteria at room temperature. The idea was to use it as a genetic tool to study temperature-dependent regulation of O-antigen. O-antigen is regulated such way that it is very abundant at room temperature and 37 much less in this enterocolitica O3. And so the idea was that the phage receptor of this one would be blocked by abundant O-antigen at 37, and we wanted to use this phage to select for regulatory mutants. It was very remarkable that this formed very tiny plaques on the soft agar, and the host range was kind of interesting. It can infect many different serotypes of enterocolitica and Yersinia intermedia, and then also Yersinia, now it is called similis, serotype 9. And here you can see that if you have very tiny plaques, it is a kind of indication that the phage might be big, and this is really true. So here you can see Yersinia bacteria and 3 phage particles there. This is another one. Here we have T4 on E. coli, and you can really see that it is quite big bacteriophage, this one. Cryo-EM shows the head, tail fibers, and then the contracted tail sheet here. Without going into more details, we have then used this cryo-electron microscopy to get structure and capsular organization of this, and it looks very much like the phage KZ. It has T27 lattice, and the dimensions are very close to the same. Here you can see the tail and the head diameters and lengths. It lyses the bacteria in about 1 hour, and the burst size is about 74 particles per cell, and it is lytic. We were then, of course, interested in what is the receptor on the bacteria, and to make the long story short, it is lipopolysaccharide, and in enterocolitica serotype O3, the bacteria express 2 types of lipopolysaccharides on the surface, and the one type is decorated with the O-antigen, which is a homopolymer, and this podovirus actually is specific for the O-antigen. And then the other type of lipopolysaccharide is substituted with the hexasaccharide, which we call outer core, and our R137 uses this as a receptor. And as you can see, the phage-resistant mutant is missing this band. So this is the hexasaccharide difference in the silver-stained doc page, and this was confirmed by complementations and so on. The hexasaccharide structure, we have solved that, and the gene cluster that actually direct the biosynthesis of this hexasaccharide is here, and in a transposon insertion library where we picked up these R137 resistant mutants, we had a transposon inserted in the WZX gene. So making mutants into the different genes in this gene cluster, we could get truncations in the outer core, as you can see here, and all these 3 truncations still bound the phage. So the phage could infect even the M mutant, meaning that the minimum receptor structure is this 3-saccharide. So now then I said that it has a different host range, broader host range, and we also isolated phage-resistant mutants from this serotype 50, 25, 26, 44, and in all cases you can see that there is a difference. This is not very beautiful gel, but it demonstrates the differences between the wild type and resistant, wild type resistant, wild type resistant, and so on. And then, of course, you start to think that these LPSs should be similar because the bacteria are infected by the phage. We determined the structure of this serotype 50, and it has no resemblance to the outer core structures, not similar to the outer core. We also studied this similis 09. Here we have wild type similis 09 with the ladder-like O antigen structure and phage-resistant mutant, which is missing then the O antigen. The O antigen structure is here, and that is neither similar to the outer core. So here we actually start to think that there are two possibilities. These different lipopolysaccharides might take similar topology so for the phage receptor binding protein, but then another possibility, of course, is that there are several receptor binding proteins in the phage. We will see. This is something that could be studied by this method that was developed in Christine's laboratory to pick up this phage receptor binding protein. Okay. What about the genome? Pulse field said that 270 kb. We tried to clone this, so this is sequencing kind of in the classical historic time. So we tried to clone the DNA into book 19, but what we did, we only received deletions, and restriction enzymes could digest the DNA, but some did not. So we thought that perhaps there are nucleotide modifications. And yes, we found out that the DNA, in the control DNA, as you can see, we have here C, G, T, and A, but T was missing completely from our phage DNA, and we had a new peak here, which was then shown to be deoxyuridine. So that is rather interesting. There are not so many other phages which have similar nucleotide composition. So how does this take place? Here we can see that this is the pyrimidine metabolism, and here we have DUTP and DTTP, and these are the kind of important crossings which might be involved in this system. And most likely we have here thymidylate synthase, and the phage would perhaps like to inhibit this reaction so that the UMP would be then directed to the UTP. We don't have any clue if we have an inhibitor for this thymidylate synthase. This has been, I think, identified from this bacillus phage. So we thought that it would be interesting to sequence the genome and identify these DNTP genes which are involved, and perhaps the enzymes and the inhibitors. We would also like to know that at what time point of the infection this change from DUTP to from DTTP to DUTP takes place, and there we have some preliminary results that this is very, very rapid. Already before 5 minutes of the infection, we start to see the kind of the decrease of the DTTP concentrations. RNA sequencing could perhaps give us some information, and we have some data waiting for analysis, and biochemistry is not done yet. So back to the sequencing, because of this DUTP business, we had to use then UNGDUT mutant E. coli to allow cloning, and we made a library and sequenced some of that, and then used primer walking to complete the genome. So I have more than 700 specific primers for this, and I think that to get this sequence out, I paid 50,000 euros or so, and it took a few years altogether to finish the sequence. Okay. 270 novel sequences, but we only could identify 5 genes which might express some proteins which are involved in the nucleotide metabolism, and this does not really explain how the bacteria make the switch. Okay, here is the genome. It came out as circular form, and as we have heard, very few DNA phage is really circular. So where are the ends? This is the BAL31 digestion analysis, and you can see that there are 2 bands here which are not predicted from the sequence. So they might represent the ends of the phage genome, and when you do BAL31 digestion prior to the KPN1 digestion, we start to see that these bands disappear from the restriction pattern. So most likely representing the ends of the genome. With SPH1 digestion, we could get an extra 6 kb fragment, and together with this, we could deduce that one of the ends should be around there in the map. And indeed, if we could map it exactly there by using a primer which is specific for this region, and it reads the sequence, and as you can see, when it has 2 templates, the sequence is higher, and then here it drops to half, and the SPS1 fragment also ends exactly there. So the left end of the genome was located exactly there. The right end we have not been able to locate exactly. Some words about the proteomics. We did some mass spectrometry analysis of tryptic peptides, the isolated peptides, and then from the whole phage particles, and could identify altogether around 70 structural proteins from this phage particle. And what you can see here is that one of the proteins, this 99 here, is similar to RNA polymerase beta subunits. Meaning that perhaps it delivers parts at least of the RNA polymerase to the bacterium when it infects. But this we have not demonstrated that yet. So one of the proteins resembles the Brohead protease of T4 bacteria and other ones, so indicating that some of the head proteins might be processed during the maturation, and if this is so, then one would expect that in the mass spectrometry analysis you might not see those peptides that are from the eliminated part of the protein. And indeed, also this head protease is known to be auto cleaved, and the cleavage site is this has a consensus sequence like this, and with this GP79 we could find a very nice consensus sequence, and the peptide coverage of the protein was in the N terminus, and the C terminus was not at all represented by the peptides. So kind of suggesting that this is really the case. And then of course this protease has targets, and we found by analyzing the peptide coverage of other proteins that it might be that these gene products which are listed there are also processed, and you can see I have here 198, 199, and here is 204, and then 206 and 275. So all these carry the consensus cleavage sites, and most probably are then processed. Some words about the promoters. So the RNA polymerase subunits we could identify. So this one, this 99 was present in the phage head particle, and these which are similar to domains of beta prime subunit are also present in the particle, and then we have 3 others which resemble RNA polymerases, and so meaning that it might well be that the phage has its own promoters. We used the program Rob Fire to look at this genome, go through the genome, and we could identify this kind of structure sequences which might be the promoters, and they were located very conveniently in intragenic regions. So most probably these are really promoters. We tried to, actually we constructed reporter plasmids carrying these promoter regions, and then infected the bacteria carrying these reporter plasmids with the phage, but in order to confirm that these would be the promoters. But the problem was that when the bacteria were infected, it cleaved away the DNA. So what instead of having promoter activity at certain time point after the phage infection, all the light disappeared from the bacteria. So we could not use this approach. So these promoters and terminators are, you can see them all through the genome, and we could identify perhaps there are 40 to 60 different operons, transcriptional units in the phage. Okay, in summary, we have identified this lytic Yersinia phage. It infects bacteria which has the lipopolysaccharide receptor. It has a genome of exclusively deoxyuridine instead of thymidine. We characterized this proteome and modeled the structure. Altogether 367 putative open reading frames, and most of these open reading frames lack homologies. The genome has been organized to 40 to 60 transcriptional units, and it has the T27 lattice. So this is my first possible stop, but I have still 2 minutes, so I could shortly show some of these genomes. So here we have 6 genomes against different, so we have myoviridae and one Syphovirus and a couple of podoviruses, and so this is that R1RT which I mentioned earlier, and this is the Syphovirus, and here are some small details, and perhaps I can just mention that we could isolate the bacteriophage which is specific for Yersinia pestis from the sewage in Turku, and the plague has been last time in Turku in 1700. So either there are very, very long-living pestis strains which are residing in the sewage in Turku, or then this phage has some other host which cross-reacts with pestis. EV1 is another pestis-specific phage, also from Turku. R801 is podovirus, and here we used LPS mutant strain, so this one most probably recognizes deeper parts of lipopolysaccharide in the Enterocoletica O3 lipopolysaccharide. And this is serotype O8 specific, O antigen specific podovirus, and then we have O8 specific deeper deep RAF mutant podovirus. This is not a podovirus, this is this Syphovirus, and this R1RT is myovirus. So now I came to the end. Thank you very much for the attention.
There's time for a couple of quick questions. So one facetious comment that Pestis phage is just downstream of where your lab used to be. But beyond that, so Fire 137, I can't remember, it presumably also contains a uracil N-glycosylase inhibitor and also a DUTPase inhibitor.
We have not identified any of those from the genome.
From the genome, okay.
Yeah, so in that sense it is not similar for the Bacillus phage.
And then one last comment, you've got a precise left end or whatever and a fuzzy right end. That turns out to be not uncommon, and I think it's a slop in the way that terminase packages. So it's not unique, and it may be a genuine result that you've got a sloppy right end.
Thank you. That is good.
Any further questions?
I noticed in the annotation of your big phage you had a toxin-antitoxin locus. Is there any information on that, the nature of that?
No idea, no. Okay.
Okay, thank you very much, Mikael.